Thanks for having us. I'm very excited to be here. And I would like to talk to you about our platform for effective antibody drug discovery against difficult targets. Now, I would like to start with the basics. Antibodies are proteins produced by our immune system in response to foreign substances, such as viruses or bacteria. Our immune system is capable to produce antibodies with exquisite selectivity in response to these cues, which allow us to defend against infection. This, along with the ability of the antibodies to have long half-lives inside the human body, have made antibodies an interesting target for drug development and discovery. Antibodies are a big market, a multi-billion market that is expected to grow in the upcoming years. Now, the process of antibody discovery relies on the use of a soluble target. This soluble target must retain its stability and also its disease-relevant properties along the discovery process in order to be effective. Using this, technologies such as hybridoma or antibody display have been very, very effective discovering antibodies against these soluble targets. However, there are serious limitations when it comes to discovering antibodies against some targets, and particularly when we care about function, not only binding. In the case of viruses, we don't care only about the binding of the antibody against the virus. We also care about the ability of the antibody to effectively block the infection, which is the important part. Because of that, we need to use either live viruses or pseudoviruses in our assay. Next, we need to express our antibodies of interest. That means that you need to express dozens, hundreds, or even thousands of antibodies in an antibody drugs campaign in order to have a few hits, if any. Then, with your antibodies and your pseudovirus, you can go through a multi-well assay that can last a few days that will tell you which of your antibodies in your sample are actually neutralizers. This process makes antibody discovery pipelines very slow because in order to be competitive, you need to use capital-intensive facilities, such as robotics. Now, this is a big problem because we need technologies to discover new antibodies fast. For example, for new pandemics, we all are aware of pandemics. When a new pandemic comes, we need to quickly screen what's immune response against this new virus. And also, we need to quickly screen vaccine candidates. And we can do it quickly with the technology we are going to present. Also, in the case of membrane targets, we are limited by the stability of the protein. Membrane proteins really like to be anchored to the membrane, especially multi-pass proteins, such as the one shown here. Now, assuming that you are able to have your multi-pass protein in a soluble format, which is very hard, then you need to raise antibodies. Most of the time, people will use a research animal, such as a mouse. The problem is that these proteins usually have low immunogenicity. So that means that the research animal rarely will generate antibodies. Assuming, again, that that worked, so this is another bottleneck, you again will start screening for antibodies that bind. But with membrane targets, again, we care not only about the binding, but also about the ability of the antibody to either activate or block the function of the target, depending on the therapeutic field that you're working in. Now, I cannot overstate the importance of membrane targets. There is a family of membrane targets called GPCRs that are being targeted by 40% of all approved drugs in the market. This is a huge market. Now, the thing is we have the ability to change the therapy and the way we treat patients with antibodies, but we are lagging because of these limitations. The technology that we are developing overcomes these limitations by miniaturizing this antibody discovery process. By testing thousands of antibodies in a single sample, we are able to reduce capital costs and increase the speed of antibody discovery. So I would like to show you an example. In this example, we mixed 3 antibodies. The majority of the antibody shown in black is an antibody that doesn't neutralize HIV, and the minority of antibodies are able to neutralize it. And you can think of this as a model of how immune responses work. When you have a sample, most of your sample will be antibodies that are not relevant for you, and only a small amount will be relevant for you. Now, after using our technology, only the antibodies that effectively neutralize HIV are able to enrich hundreds of times as compared to the irrelevant antibody, which remains unchanged. Now, this is a proof of concept, and currently we are testing our samples with thousands of antibodies to effectively discover the needle in the haystack. By using our technology, we can decrease significantly capital costs. Antibody discovery campaigns are currently very expensive, but by miniaturizing this process, we can decrease those costs. Only using 1 or 2 trained technicians, you can screen thousands of antibodies in a single sample and as a consequence of that, you can decrease the cost per antibody screened. I would like to introduce our team. As I mentioned before, I work along with Dr. Brandon Nikoski of MIT. We also work with a graduate student from our lab, Matias Oliveira. I would like to thank also the Despande Center and our lead catalyst. We're very interested in growing our team, especially on the business side, but also on the research side if you are interested. So feel free to come to our poster and ask us any questions. Thank you.